Hi, today I am going to present one of the most interesting topic, which is basically for the new advancement in the multi-omics data analysis and their real application, or you can also say the case study. So here you can see the different titles. For example, gene expression and mutational profiling to pathway crosstalks. Then you can see from multi-omics data via gene expression, mutational analysis, and pathways enrichment analysis, and network biology to systems pharmacology. Then there is third title, that is GECIP toolbox, which is titled as an approach for the systems level understanding of prostate cancer from high throughput data integration to pathway modeling and simulation. Then there is another screenshot which you can see titled as the same, an approach for the systems level understanding of prostate cancer from high throughput data integration to pathway modeling and simulation. So this is one of the software which I had developed in 2021, and this was published with the real application in cells. So this is basically a kind of software which I have named it as GCIP Toolbox, which is the replacement of EPA software, or which you also call it as Ingenuity software, which is kind of highly paid software and commercial software. But this software fulfills the goal of what EPA or Ingenuity software fulfills. This fulfills more than even that in terms of the prediction, and it's relevant to the experimental validation. So here I am going to teach you few things which will help you to understand and analyze the multi-omics data analysis and their integration at multiple levels. Then pathway enrichment analysis and pathway crosstalk. And finally, you can also think or understand the network level understanding and analysis, and to up to the systems pharmacology. So here we are combining basic bioinformatics, systems biology, experimental data validation, and mathematical modeling and simulation. So now let's proceed. So if you are interested, then you need to, in such kind of real application, first thing you need to understand is detailed workflow, how the workflow would look like. So the workflow, here you can see clearly for in case of gene expression analysis, when you receive the raw expression files for different data sets, then you go for its analysis to convert it the real gene expression value, or if it is for the mutational purpose, then you go for the genes, prediction of the genes with different types of mutations. Then you go for differentially expressed genes or differentially mutated genes, and finally to pathway, their impact on the pathway or pathway enrichment analysis. Then you, in this case, how this software could work is basically you need to go for the DEX list or differentially expressed genes list and the enriched pathways list. Then you prepare different networks and go for some factors to be included in pathway crosstalk. So, and then some of the simple mathematical equation, how you are going to calculate the degree of pathway crosstalks. So this is what in simple it may look like. And if you are interested further, then you need to understand this workflow, which was detailed workflow in summary. So in summary, if you want to understand, you have different cases, then you have down-regulated genes, up-regulated genes, and the total differentially expressed genes list. Then predict the upregulated pathways, downregulated pathways, and the overall up and downregulated pathways. Then you go for crosstalk degree calculation and classes prediction, then pathway interaction for the corresponding genes. Now, if you want to understand the illustration for TCGA data set, if you are looking for, instead of your own data, if you are looking for the biggest cancer database, that is TCGA data set base, where you can select the ovarian cancer data set, then how it may look like. You can go for different types of data set, like the copy number alteration, DNA copy number data, and then, like in this DNA copy number data, you can go for consensus reason, some networks, then significant subnetworks, and go for the clinical survival lists. Then it comes to the KEGG pathway. So many people do not know what are the categories or classes of KEGG pathways. In general, they are divided into 7 different classes, but there are mainly 4 pathways classes. One is metabolic, then genetic information processing, environmental information processing, cellular processes, and organismal systems. Human diseases and drug development pathways are basically the derived version of these 5 classes of pathways. So there are more or less 232 pathways, total pathways, which are grouped into these 5 classes of pathways. So for the purpose to develop our software, we have excluded the repetitive pathways classes, that is drug development class and human diseases class, and considered these 5 main classes only. And now, if you want to understand them, how these 5 pathways are here, then connected with each other, of course, because they have the proteins as the components, pathways components, or genes as the pathways components, then metabolism, genetic information processing, environmental information processing, cellular processes, organismal systems. Here you can see the lines connecting all the pathways with one another. So every pathway class is connected with each other. Now, you can also think about the individual pathways. Instead of individual pathway, you can also think how the metabolic system is interacting or disturbed in particular kind of human disease with respect to genetic information processing or organismal systems or cellular processes or environmental processes. But mostly the people are interested at the individual pathways level. So how the crosstalk outcome may look like when you are thinking about the pathway crosstalk, then this is just sample example for different crosstalk degree or scores. So for example, if you have the scores, maximum if you are going to apply, then 0.7, that is the maximum. Then you have less number of pathways interacting with each other. Then there is 0.5, you have more pathways interacting with each other. Then 0.2, 0.1. So depending on the interest, what are the pathways you are interested in? Is it coming for the maximum crosstalk score or degree, or if it is coming at slightly lower crosstalk scores or degrees? You can decide, and based on that, you can go for further analysis of the crosstalks. So here, now we are coming to understand the network. For example, if someone is just interested only in the biological network or gene networks or protein networks, then how they can proceed. So you can think about, first thing is, all these differentially expressed genes should be plotted with their respective expression values as a heat map. Here you can see in the middle. So this is, for example, enriched common pathways. Here it is the p-values, which is basically p-value is used for all the pathways. But you can think similarly for all the genes also, their gene expression values and fold changes, and of course, if required, but uh, p-values may not be that impactful to show in the heat map for basically differentially expressed genes when you are showing the gene expression values and fold changes simultaneously. So you can think about the first thing is immediately you need to look for the heat maps for the expression values or p values or fold changes. Then you can think about the network as you can see here in the left side and try to focus on the beautification part also or visualization part, presentation part, so that it should attract immediately the attention of the audiences. Now you can proceed further how you can think for a simple research paper. So, for example, how many differentially expressed genes in figure A, you can see the workflow. So, workflow is always helpful when you are doing some research work. It is always helpful to present the workflow. And then B is representing the total number of differentially expressed genes in prostate cancer and breast cancer and ovarian cancer. And if you have more data sets, for example, here three data sets, then in all the different data sets, how many genes were differentially expressed. Then you can think about the Venn diagram when you have different cases or conditions. And finally, you can think also about the heat maps. So this is the simplest way to prepare your easy or simplified manuscript or research paper. So here the important thing is from where you are getting the data set. For example, for gene expression or mutation data set, then CY portal is the first thing or TCGA database where you can look for it. But of course, gene expression omnibus is the common place from where mostly the people used to download the data. In addition, there are some disease specific databases also. For example, PCGA is giving you only cancer data set, but gene expression omnibus gives you all types of data sets which are available. Then it comes for the NEF, like the kidney disease or diabetic patients samples or data sets. Then nephrosic is also one of the options. Then there are many more types of data sets which are available for disease specific conditions only. Now you can think further how you are going to proceed further if you want to add something new for the similar analysis for the visualization purpose. The best way to present your result or outcome in graphical way, then you can think about some pie chart or even the heat map could be expanded or magnified. So for example, here you can see, but here also this heat map could be beautifully plotted by using some kind of white lining in between. For example, here you can see the white line have been inserted, which clearly segregates or separates the three different colors for three different conditions. And the pathways here, all the pathways be in three different conditions: breast cancer, prostate cancer, ovarian cancer. Now go for the next part to understand much slightly different way. For example, if you are looking for the target proteins or biomarkers and then drugs. So basically in case of drug, then how it would look like. So there are a number of ways to present your data while you are giving the presentation or presenting your data for research paper purpose or research manuscripts. Here, these are the best way to represent the different pathways, different genes with their heat maps, and in a well-defined way. So it is easy to understand for everyone. For example, here we are showing the PI3K AKT signaling in first data set, that is Grasso data set, is showing some different color for the minus log 10 of p values, and then it is much higher color in like the deep red color in GSE 8218 data set, but cancer is prostate cancer. And then it is Third data set for the prostate cancer is 1431. Here again, it has similar kind of significance value or p-values with the Grasso dataset. But in case of GSE 8218, it shows the maximum significance. So here, this PI3K AKT is showing the highly enriched pathways in irrespective of the data set. They are showing the maximum significance. That means this particular pathway is highly altered when there is prostate cancer. So this is how you can interpret these heat maps also. So you need to learn the best way of presentation and best way of explanation while you are writing the manuscript. Now, if, for example, here I have presented the total number of differentially expressed genes in different data sets. For example, in Grasso, total number of differentially expressed gene is 390 for prostate cancer, but in GSE 8218, it is 350 differentially expressed genes, and in third data set, GSE 1431, it is 257 differentially expressed genes. But when we are looking for the total number of enriched pathways, then in Grasso, it is 27 pathways, and then in GSE 8218, it is highest, that is 45 pathways. Then in third case, GSE 1431, it is slightly lower, that is 22 pathways. What you are seeing here in case of Grasso, there is maximum number of differentially expressed genes, but the enriched pathways are not maximum. It is showing maximum for the second data set, which has the slightly lower number of differentially expressed genes than Grasso dataset. So it means that in case of Grasso, there are more number of genes differentially expressed or altered, but when it comes to their functional impact, then they are showing comparatively less number of pathways. So this is how you can conclude or interpret. Now, it comes the best way of presenting the prostate cancer pathways crosstalk. So here, this is first data set where you can see that is 8218, GSE 8218. What you are seeing here, there are more number of pathways which are showing the crosstalk with each other. But there are 5 pathways which are in the center. When you are looking for the second figure, here also there are 5 pathways which are common with the first data set. But when you are going to the third network for the third data set, then here you can switch. See, all those 5 pathways are more or less present here, which means that irrespective of the data set, these pathways are promoting the maximum pathway crosstalks. So in case when you have good analysis, then irrespective of the data set, the final conclusion should remain the same. As you can see here in the first net crosstalk, the maximum pathway crosstalk could be seen here for MAP kinase signaling pathways, RAP1 signaling, focal adhesion, PI3K AKT, adrenergic signaling. Similarly, these pathways you can search in all the other 2 pathway crosstalk figures, and you can easily find them. And it is not just you can easily find, but they are showing the highest pathway crosstalk or pathway interactions. Now, the conclusion here you can say, irrespective of the data sets, in case of prostate cancer, PI3K AKT, MAPK signaling, RAP1 signaling, and this, like, adrenergic signaling, they are basically appearing as showing the maximum pathway crosstalk. And that is the conclusion. Now, if you want to go for much deeper analysis, then you can make a network of not just pathways, but also with their genes or proteins, which you can see here in figure number C. Those pathways, which you are seeing in figure, left side corner figure, pathway network, these 11 pathways are having these particular proteins, which are the source of pathway crosstalk. So then you can also, this is like individual way of presenting. First is pathway network, then another is the pathway component network or PPI networks, that is protein-protein interaction network. But I will show you how to merge them together in the next slides. So then figure B is showing the crosstalk scores for all the pathways, 11 pathways. And figure C is showing the last bottom line figure here, all the 11 pathways with those proteins or genes which are like just in how many pathways they are present. For example, here AVI2 is present, the first gene, in only one case, that is regulation of actin cytoskeleton. But ACTN2 is present in two pathways, that is focal adhesion and regulation of actin cytoskeleton. There are other genes, with many genes which are present in 5 pathways, 6 pathways, 3 pathways. So this has certain meanings. So what does it mean when one gene or protein is shared by 5 pathways, 3 pathways, or 1 pathway? It has some biological meaning. Now I am going to explain you these combined pathway network and pathway components networks. So these different colors are representing, the groups of different colors are representing a specific pathway. For example, this cyan color is representing exclusively the regulation of actin cytoskeleton, and the number is representing the pathway index or pathway number. So 5 is for regulation of actin cytoskeleton. Then in the same way, you can go for other colors. So all these 11 pathways have been represented here. One is representing PI3K AKT. But when you are seeing one gene or protein with 4 colors, 5 colors, that means we are trying to show that this particular GSK3 beta is shared between these pathways. That means this protein is not only the component for thyroid hormone signaling, or for this hippo signaling, or for the focal adhesion signaling pathway, but they are for all these 4 pathways. They are present in all the 4 pathways. So that increases the significance of this particular protein, GSK3 beta. And that is what is known for GSK3 beta, that it plays crucial role not only in cancer, but also in neurological disorder and diabetes. So this is how you can interpret the significance of this. And here you can see in the prostate cancer, this particular gene is altered. So that means this, when there is alteration in GSK3 beta, it is not only going to affect one particular function, but it will affect 4 different biological functions. Now, this is primary consequence, but there are secondary consequences also when this particular gene is connected with another gene, and that gene is representing the other pathways. So this is grave consequence of the alteration in the GSK3 beta gene expression. Now, the real application of system biology, when you have analyzed all the networks of the genes and the pathways, then you need to think about the experimental validation. So here we had the prostate cancer patient for which we have collected the biopsy samples, and then we tried to predict some of the mathematical modeled proteins. For example, here predicted the ERK signaling in case of prostate cancer by using this small components of signaling pathway, which you can see here in figure A, and then how it looks like for different input signals. Here you can see for different input signal, the different responses of the activated ERK protein. And the same thing we observed in case of figure C for human biopsy sample for prostate condition. And then you can see in figure E that is representing the docking outcome of these proteins, like for example the top ranked network proteins for GSK3 beta, HIF1A, YWHAE. These three proteins were the top ranked in terms of network level understanding, and we targeted with the natural compounds, that is apigenin, quercetin, and resveratrol. And what we observed here, that apigenin and quercetin is showing the maximum binding affinity with these top ranked proteins. So we can conclude from here that these top ranked proteins are showing the potential for considering as the main drug targets or biomarkers, and apigenin and quercetin will be one of them. Now you can think about the selected pathway. You need to understand while you are working on the such big project. So this is basically what you saw here. This is one of the finest example of systems pharmacology. So you need to understand how the pathways are in one pathway. The different proteins are connected with each other, which protein is activating, which protein is deactivating, and how they are regulating different proteins in one pathway. Now, someone can ask that in addition to these binding affinity, how we are going to analyze. So here it is very simple. You can use any tool which are freely available. And then you can go for docking, which you can see here on the right side top. There is drug and the target protein. Then third figure, which is on the bottom line, here you can see the binding sites of the protein and the drug components. So this is best way to present the protein ligand docking analysis results. Now, when you are talking about prostate cancer pathways and their visualizations, crosstalk visualization analysis, you need to think deeply. As I mentioned, find the best way of presentation and visualization so that people before reading the text can understand from the graphical presentation itself. Now, this is the deeper analysis for the prostate cancer network components, and these are one of the main point which you need to analyze in systems pharmacology. Of course, I can understand many people are not aware about pathway crosstalk, but you need to think how to present these or how to analyze the pathway crosstalks. So prostate, this further visualization could be done in another way. For example, here pathway network, then gene components or protein, the components of the pathways, then their crosstalk scores, and then the degree of all the genes or the proteins present in this particular network. So for example, here you can see the how many proteins are with the highest number of degrees, how you are going to present. These are really worth while you are working in network biology or systems pharmacology. Then there are some more things called connectivity, adjacency matrices. These things you need to understand. These are primary things which you need to understand and plot them together. Now, if you are, this is another way of presentation. Of course, it is not that attractive, but this is just to show in different types of cancers for different values of the crosstalk scores. We have plotted the crosstalk, pathway crosstalk. Then Next is the last part, which you need to understand these days. Most of the journals, they ask for the graphical abstract. You also need to be expert, whether it is review paper or research paper, how to present the graphical abstract for your entire work in just one figure. And this is how you can make a beautiful figure to present the graphical abstract. Graphical abstract means with the figure, you are summarizing the entire work from analysis to the conclusion. And that is what you can see here. So here you can see the analysis, then method, then results, then final conclusion, where we are looking for the final goal of the work. So now, finally, I am coming to the point in today's lecture. What I was trying to conclude is multi-omics data analysis and pathway level understanding and multi-level data integration. Then network biology application with the real case study, systems pharmacology with real case study. Finally, with this work, today's talk, you will be able to easily develop a research paper or the manuscript. So thank you very much for listening my videos and watching my channel. Thank you all.